Teachers in general are my favorite audience. I was a teacher for 2 years. I taught 3rd grade, 5th grade, 7th grade, and high school science. I taught botany, which is a real pleasure and unusual for the school system that I was in, which is in Brooklyn. I also taught physics, chemistry. I was sort of a jack of all trades, science teachers. I met my husband when I was teaching. He's also a teacher. I now have 2 daughters. They're 10 and 7, and I live in Newton, and they're in our local public school system in Newton at Underwood Elementary School. So just all in all, I think it's just I mean, I used to go to teacher outreach, like anything I could get my hands on. So I'm completely I'm resonant with this audience. And I used to love it myself, so I'm happy to sort of be on the other end of it now. So I want to start off by telling you a story. I also know that, by the way, that teachers are some of the worst audiences ever because they like to talk to each other and they actually want to be teaching, they don't want to be students. So I'm completely fine with that. And also, if you have questions, you can shout them out in the middle. I really don't mind being interrupted. I have many things to say. We have an hour. I'm not going to finish everything that I have to say. So I'll just talk until the time is done and try to tell you about why I think biodiversity is important, but I'm sure we'll all get a lot more out of it if you have questions, if you ask in the middle. Don't wait until the end because we may run out of time. All right, so why is biodiversity important? Well, there are lots of hypotheses, I suppose, about why biodiversity is important, but here's another one for you. So last fall, I live in Newton. I work with a mushroom that's the death cap mushroom. It's reputed to be the world's most poisonous mushroom. In fact, if you eat it, you do die. I work with a hospitalist. A hospitalist is the person, when you present at the hospital, who manages your case. So an MD. So I work with an MD out in California who a while ago, several years back, was confronted with a family, an immigrant family, Southeast Asian family, that had eaten some mushrooms and they were sick, and many members of the family were on the verge of death. He investigated and found an experimental therapy that's available in Europe where it's available because there's just more of a culture of eating mushrooms in Europe. It's not available in the US because to get FDA approval for the drug would cost an awful lot of money, and the company doesn't want to go through it. They wouldn't make any money off the drug because there are not that many poisonings here. So he got permission from the CDC to have it couriered in. He treated the family. Long story short, they actually recovered on this experimental milk thistle therapy. So for that reason, he's gotten permission from the FDA to do an experiment of sorts here, but it's unethical to really, you can't do a controlled experiment. If someone's eating a mushroom, you can't say, I'll give you the milk thistle therapy, but you I won't. So it sort of has its own guidelines, and one of the things that he needs to know when he's involved with the poisoning is what exactly the mushroom is that poisoned the person, because he needs data for the National Institutes of Health to prove to them that this therapy is effective against what he says that it's being used for. Okay, so last fall, I got news of a poisoning in my hometown, Newton, Massachusetts, and the poisoning ended up at Mass General in the liver transplant unit because this fungus destroys your liver. It actually stops RNA polymerase II from working, so it stops protein translation, and actually molecular biologists use it for that reason. They actually buy it from Sigma, and every once in a while I get someone asking me for it for their molecular biology probe. So they ended up at Mass General. There happened to be an Irish guy over at Mass General who's head of the liver transplant unit. So he had familiarity with this, and he looked at the poisoning and he said, it's consistent with an Amanitan poisoning. It must be Amanita phalloides. So at that point, the hospitalist from Santa Cruz got involved because he's the only one who can dispense this experimental therapy, at which point I got involved to try to identify the mushroom. So I get on board with this guy who's, he's already been administered the drug, but they want to know, sorry, I wonder whether I can not make this go away. So they want to know what it is that he ate. And so I ended up all over the news telling people that Amanita phalloides does not grow in Newton, Massachusetts. It's my hometown. This is my species. It's what I work with. And I know that it's not in Newton, Massachusetts. And the general message, which I tried to get across, which nobody put me on the press anywhere, but I kept saying to people, people just freak out over poisonous mushrooms. And I kept saying to people, it's actually quite okay to eat mushrooms, but know your biodiversity. If you can tell cilantro from parsley, you can tell an edible mushroom from a poisonous mushroom. All right. And the reason that I said it's not an Amanita phalloides is not only because I've never seen Amanita phalloides in Newton, but also because I actually talked to the victim in the hospital, and he described the mushroom to me. He was Ukrainian, and he thought, and he and his mother collected it. And so he thought that it was phalloides, and he gave me this long description. He gave me the Russian name. I happened to have a student who spoke Russian. We looked up the Russian name. It had nothing to do with Amanita phalloides. Okay, fast forward 2 weeks later, where I meet the victim at his home in Newton, and he takes me to the patch of forest with my 2 daughters, by the way, which he was really nervous about. But I was like, don't worry, they go on lots of mushroom forays. They know not to eat Amanita phalloides. But he's just, unlike you. But so he took me to the local tree, and lo and behold, I had to eat crow because there was Amanita phalloides, which first of all taught me that I have a cardinal rule about not identifying things over the phone, because I really don't like to identify things over the phone. But in this case, I did because of the circumstances, involved. And it goes to show that this person, he couldn't, he was looking at something and he couldn't tell me what it looked like. I think what he was doing was giving me over the phone the description of the edible species that he thought he was eating, even though when he actually showed me the mushroom, it had nothing to do with that Tricholoma species that he thought he was eating. It was green, everything about it. So there are so many compelling reasons to think about biodiversity loss and global change. We're living in an era of global change. I'm going to talk a lot more about that later. But it's just, I think, a story that's really indicative of how we don't know anymore so much about what's happening in our natural world. We aren't trained, we can't see that green is green somehow, and that actually pale cream with olive striations is pale cream with green lines running through. So it's just another story about what it means to know or not know your local biodiversity. And this is just one of the stories about it. Okay, so with that, I'm going to go to the actual substance of my talk. So, and these are things I think about all the time. So I use the word I a lot, but it's a lie. My lab is a team, it's a we. We work a lot with a lot of different kinds of people. This is Franck Richard. He's now has a permanent position back in France. He's a French postdoc in my lab. Ben Wolf, who's sitting up in the lab right now. They're very happy because, I don't know if you can see, there's a little knitted gnome. One of the amateurs that we work with dyes wool with mushrooms in different colors, and then she knits it into these little gnomes, and she's was very happy. That's my technician Hugh and his wife Patty, and that's my daughter Penelope. This is just from a party at our house. But these are the people who really did so much of the work that I'm about to tell you about. Okay, fungi. Maybe when you took high school, maybe you learned a paragraph or so about fungi. If you did, what you learned is that they're plants. But it's not true, it's not true, right? Fungi are more closely related to animals than they are to plants. The first evidence came from DNA sequence-based approaches, but then later on people have done careful morphology and proved the same. So how many fungi are there? It seems like, in thinking about biodiversity, one of the most basic questions that you should be able to ask me is, how many species are there? And I should be able to give you an answer, but I can't really. Why? Because many fungi are cryptic. They grow underground. You only see them when they produce these beautiful mushrooms or other reproductive structures. It doesn't happen all the time. It's not always obvious. There are not that many mycologists on the planet, actually, so there are not a whole lot of people who are invested in studying these organisms. And so we don't really know how many species there are. We know that we don't know how many species there are because we discover them all the time, and it's commonplace. So another one of my themes about biodiversity is that I do think that there's a tendency to think that biodiversity lives somewhere else, namely the tropics. We hear all the time about how biodiverse the tropics are, and I think that does something really awful. It makes issues of biodiversity someone else's problem. But if you work with fungi, you can't get away with that, because in fact you discover new species all the time. I work with the genus Amanita. It's one of the most charismatic genera of fungi that exists on the planet. The first year I was here in 2000, actually it was the summer of 2006, I don't know if you remember, it was a deluge. It was one of the rainiest summers on record in New England. We were bringing new Amanita into the lab all the time. So one of my messages to you is that we don't know a lot about fungi, but biodiversity is not somebody else's problem. We have a local biodiversity. We are in stewardship of our local biodiversity. There's a lot of it, and there's a lot of undescribed biodiversity. Naming new species, discovering new things is not something that just happens far away. So This guy named David Hawksworth went around the world, and I'll introduce you to some of the players here. So some of these you'll know really well, like truffles. Probably everybody has heard of a truffle, and some people have eaten truffles. Mushrooms, generally, this is Amanita manicata. It's growing on the beaches in Hawaii, thus the lovely palm trees. Lichens, these are also things that are familiar to people. Yeasts. So if you think about how you interact with fungi in your daily lives, which you may not think about, anybody who eats bread or drinks beer or uses soy sauce is relying on a fungus. Any product of fermentation that involves a yeast is a fungus, right? Tempe, many things use fungi. If you do laundry with color brightening detergents, those enzymes are actually fungal enzymes. And what they do is, you know how fabric has a tendency to pill? It makes these little round balls of fiber. These fungal enzymes go and they clip off all the bases of all those little pills. So biofuels, how do you degrade a plant and then ferment it to get the ethanol you put in your car? Actually, you do that with a fungus. So even though these are things that I think your students don't think about, there's heaps of reasons there are lots of ways actually where you interact with fungi in your day-to-day life. This is a more obscure group of fungi, the Labule-Beniales. This is a ladybug. These are fungi that are growing on the back of the ladybug. These are obligately insect-associated fungi. There are lots of them. Biodiversity is the discovery of biodiversity is commonplace. It's routine, even in New England, where you might not think it's a biodiversity hotspot. And actually, we don't know that latitudinal gradients are real for fungi, just by the way. So you think that plant diversity is greatest in the tropics, you're right. But we don't know that that's true for fungi. We have no evidence to suggest that there's any sort of latitudinal gradient in species diversity when you're thinking about microbes. And in fact, some evidence to the contrary. So we think that based on ratios of fungal species to plant species in certain places where they're particularly well studied, that there are about 1.5 million species of fungi. Although actually even this number is fraught with uncertainty. The take-home message basically is there's just a lot of it we don't really know for sure. But we know for sure that we don't have names for many things. Okay, so we know that habitat loss, or we think we know that habitat loss and invasion are major threats to biodiversity. This is a website called natureserve.org where you can sort of catalog the number of species in your local area. If you go to that website, then you can find out that just in the United States, cataloged, there are 18,400 species of plants. 15% of those are cataloged as imperiled. So we don't use the word endangered because endangered is a political word. It means something legal. Thus, it has lost some of its scientific value because something can actually be quite in trouble but not be labeled as endangered. So we just use the word imperiled. 15% are imperiled. Fungi, there are over twice as many described species, and I've just gone on a long rant about how probably there are, we know that there are lots of undescribed species. So there are at least twice as many described species of fungi in the United States. Only 0.4% of those are imperiled. So you can come to one of two conclusions. Either fungi are less likely than plants to be in trouble. That may be true. Or we just know so little about fungal dynamics that we can't even put a number onto that. And that may be true too. We don't know. So this is the system that I use to think about all of these things that I've just laid on the table. This is Amanita phalloides, the death cap mushroom. In California, where I first encountered this mushroom, this poster is put up in any place where there's a population of Amanita phalloides, and it's to warn people that this mushroom is deadly poisonous. It's printed in all of these different languages because it's quite often people coming from other countries who have an image of a mushroom that they collected and ate in Southeast Asia, in Eastern Europe, that arrive in North America, and they see something that they think looks alike, and they eat it. So this is the same warning printed in many different languages. It's a symbiotic fungus. It's a mycorrhizal fungus, and some of you may have heard that word before. So what it does is a plant never grows alone all by itself, even though you can grow a plant all by itself in a pot in the house or in the garden if you add enough fertilizer. But in forests, as you walk through them, those roots in the soil are associated with fungi. Those fungi act as networks, extensions of the root system. They capture nitrogen, which is a limiting resource for plants. They bring that nitrogen back to the plant, And in exchange, they receive carbon compounds from the plant, excess photosynthates. By the way, I published do people read American Biology, Teacher? Okay, I published an article. I was a co-author on an article in American Biology Teacher that lays out an experiment that you can do with mycorrhizal fungi in your classroom. So, and if you can't find it, email me. I'd be happy to provide it.
Which issue?
Oh gosh, I don't know.
I'll get it from you tomorrow.
Okay, yeah, you can send it around. And actually, maybe now is a good time. So my outreach focus is honestly more targeted to elementary school kids than high school kids for lots of reasons. All the reasons that people like one age versus another age. But I recently got funding, and if we have time, I'm going to try to play this, although it's a large group, from NSF. Maybe I'll just pass it around so you guys know what it is. I got funding from the National Science Foundation to print out many sets of this game that I made that's called Mushroom Bingo. So this is targeted towards third graders, but I sort of wanted to make sure that you guys knew about it. And I have money to provide a set to anyone who wants one who's a teacher. And all I need from you is, actually here all I need from you is your name. If anyone not in this room wanted a copy, they just need to write me on school letterhead. And the idea is, I'll just pass these around so everybody can look at them. The image quality will be better on the final copies. They're really pixelated on this because it was a low quality PDF that we used. But the idea is that as a teacher, you would have call-out cards that look like this. So the idea is, in a 5th grade classroom, but I think there are actually ways you could do this in the high school too. You would you would say Amanita Franchetti, and then kids would look and they would have to find that on their bingo, and then you have a rock, and you can mark it on, and then when a kid gets bingo, and I've tested this in classrooms and it's really fun and it works really well. And when I do the final version, on the back I'm going to put, this was a suggestion from one of the teachers I tested it with, I'm going to put a natural history note about the fungus so that you can say, this fungus is known to do X, Y, Z. So anyway, so if you want one, I think
They can send their names to me.
Perfect. So tell Tara if you want one. And if you use it and you don't mind dropping an email, that you used it, I wouldn't mind knowing about that either. That'd be fun for me to know. Okay, so that's something that's available. And that's all based, okay, so that's all starting with this work with phalloides. Sorry, back to phalloides. All right, so there's a story about phalloides. So remember that I told you that habitat loss and invasion we think are major threats to biodiversity, and I told you that we don't know actually a tremendous amount about fungal biodiversity or whether fungal species are in trouble. So when you give to the Nature Conservancy, you're not usually giving to the Nature Conservancy because they're preserving fungi. It's just not a part of the conservation biology dialogue. So all this became really interesting when I was living in California, and there was a rumor in the amateur community that Amanita phalloides was an introduction. So this symbiotic fungus, this thing that associates with trees, people had recorded, especially in the 1980s, that it might actually be a European fungus that was introduced to California specifically because they brought cork oaks from Europe to California. So a cork oak is it's a Quercus species where you can harvest the cork for wine bottles. And the idea was to bring in these cork oaks for the domestic wine industry in California. And actually one of the people who died early on from Amanita phalloides was a son of the Sebastiani wine family. Though I've never quite had the guts to ask them for money to support my research, but I keep thinking that someday I can write a really diplomatic letter and ask if they don't want to support some of my research. So the story was that it was a European immigrant to North America, and that was really interesting to me because at the time we had no examples of a fungus, of a non-pathogenic fungus that has been introduced and is invasive anywhere on the planet. So contrast that to what you know about invasive plants, where I'm sure all of you can think of many invasive plants just in New England alone. But we didn't have any examples of non-pathogenic fungi that were invasive, none. Okay, so this is a picture of Amanita phalloides growing in a habitat in Point Reyes National Seashore. And this picture is also to remind me to say again that these mushrooms, they're just like apples, right? They're the fruits of a much larger below ground structure that can stretch for meters or in some cases acres, actually. The very largest fungi can be miles across underground where you can't see them. So and this is the root tip. So there's a mycelium, and this root tip is the site of the physical symbiosis between the plant and the fungus. So this is where carbon is exchanged for nitrogen.
Would it be harmful, or do you have to ingest it?
You have to eat it. It's not, and in fact, every mushroom is okay to touch. So if you're wandering with your students in the forest, it's completely fine to pick up a mushroom and handle it. You have to eat it to die from it. All right, so these are the beautiful forests in California where you find Amanita phalloides. These are coast live oak forest, Point Reyes. How many people have been to Muir Woods? Maybe, okay, so that's where I work, okay? So imagine Muir Woods, that's where I work. We know from data that we have that when you walk through a forest, patch of forest where Amanita phalloides is invasive, if you harvest all the mushrooms off that forest floor over a season, 80% of the biomass is Amanita phalloides, is the death cap mushroom. So where it exists, it's really dominant. So if it really is an introduced species, one might think that it could be having impacts on local biodiversity, either fungal biodiversity or plant biodiversity. Because if it's an introduced species and it's now associating with these beautiful trees that have never associated with phalloides before, one could think, for example, that maybe phalloides is taking more carbon from the plant in California than it would from the plants it associates with in Europe, because you've disrupted a coevolutionary relationship and you formed a new relationship, and how the balance of carbon and nitrogen goes might be very different in the new habitats versus the old habitats. Okay, so the basic questions I had when I started this work were, is Amanita phalloides an introduction? So an idea, it was a rumor, an idea, it was more than a rumor, but it was an idea that's not the same thing as data. So we needed a little bit more data before we concluded that was an introduction. If it is an introduction, how far and how quickly has it traveled, and what's helping it get around? You know, how does it get around? So these are sort of some of the basic questions that I started with. Okay, so if you want to know if a plant species is native or introduced, probably you pick up a book, right? And you'll read that it's introduced to North America, like kudzu. You know, we know where kudzu comes from. We know where garlic mustard, we know where garlic mustard comes from. We know where it's growing now. There's no resource for fungi that way. If you pick up a field guide to local New England mushrooms, there are not even range maps in there. It will give you a general sense of where a species grows, but it doesn't have the sort of beautiful state-by-state picture with some areas shaded and some areas not. That's because we know so little about the basic natural history of most species, we can't do that. So literature, in the end, is not an extremely useful tool, and I'll talk more about that. And I finally had to turn to DNA to solve this problem. So literature. Here's a beautiful illustration from Barclay, you pronounce his name Barclay, published in 1860, a book called British Fungology. And I use this because it shows you that in the UK and in Europe generally, this was the conception of Amanita phalloides. So in particular, I want you to notice that it has a green cap and it has sort of brown stripes on it. So the species concept of Amanita phalloides in Europe looked like this mushroom in 1860, okay? If you were a botanist in North America in 1900 and you collected a plant and you didn't know what it was, you could dry it as a herbarium specimen. You could send it off to an expert in Europe, and they would be able to use that dried herbarium specimen to identify a plant. When you dry a mushroom, it actually loses a lot of what makes it valuable for a taxonomist. So it's really difficult for an American mycologist then to dry a mushroom down and send it to his counterpart in Europe and ask, what is it? So you had to rely on field guides. And usually North American mycologists relied on European field guides. So McIlvaine and McAdam, this is the first work of American mycology. It's called 1001 American Mushrooms, published in 1902. And I want you to notice the elf reading his toxicology textbook by the skull in the midst of this garden of poisonous mushrooms. One of the poisonous mushrooms that he's sitting next to is Amanita phalloides. I think you can tell instantly that that looks nothing like the last picture. And it gives you a sense of the species concept that people in North America were using as compared to the species concept that people in Europe were using. Why the species concept, this is another version of Amanita phalloides, brown instead of green. Why the species concept changed so much, I don't exactly know. I don't know. I think Amanita phalloides is a very charismatic species because it kills you and people sort of like to find things that kill them. I don't know, but for whatever reason, American mycologists used the name in a very different way. So here's another picture of this guy, Walter Murrell. There's so many stories about the personalities involved. This Walter Murrell ended up in an insane asylum in Florida. which is a fine tradition for many mycologists. We seem to end up in insane asylums in various places, or as department chairs. That's sort of, you have one of two options. So this brown mushroom is Murrell's idea of Amanita phalloides. Okay, that's also a problem because Murrell worked at the New York Botanical Garden, and he deposited many dried mushrooms with the name Amanita phalloides in the New York Botanical Garden. But when you look at this picture, your ability to trust those herbarium specimens is zero. All right, so the literature doesn't help. And this is Jacob Lang, another mycologist, a European one in this case, who was actually here on a religious tour, but he really knew his mushrooms. He was proselytizing about this particular religion that he was involved with, but he did actually record his mycological observations along the way. And he pointed out that when an American mycologist who knows the European species from descriptions only faces the problem whether a particular fungus is the one named Agaricus whatever by Fries is our Linnaeus, he will often be at his wit's end and may count on his buttons to settle the case. In other words, I want you to get a picture of North American mycology that's just like out there. Anything goes. Any name goes. It's really hard to figure out what's going on. But by the 1970s, when we had such things as airplanes to take us back and forth, specimens of that European Amanita phalloides, that European species concept of Amanita phalloides, were found on both the East Coast by Tanguy and Simons. I think it was Simons who's actually a priest, but then in his spare time collected mushrooms. I'm telling you the story, it's just go on. And Joe Amirati et al. On the West Coast. So certainly by the 1970s, it had arrived. Whether or not it was there before, the literature is sort of a mess about that. This is work that I did with my colleague Elza Valinga, just to try to understand sort of the current state of where Amanita phalloides is and isn't, and where we could decide it was native and where it wasn't. And the literature now makes it quite clear that it's, sometimes I get asked, how do you know that it's native to Europe? And basically we know that it's native to Europe because it was first described there in the 1700s. And but we don't actually know how far it extends into Russia. And we know it's been introduced on the East Coast because somebody very carefully in the 60s looked for the European Amanita phalloides and didn't find it and wrote it down. So we know actually that it's introduced on the East Coast, but we don't know whether it's introduced. At the time, we didn't know if it was introduced to California. And it has actually been introduced to a number of other different places, which I won't talk about very much today. Okay, so genetic detective work. So I want to make this long story short, basically. So we don't know. So basically we're in chaos. We don't know whether phalloides that we find in California is an introduction or not. The literature doesn't help us. So we have to use DNA markers. Well, what would you expect, looking at DNA sequence data, what would you expect if you had a population in California that was native and then a European population? I mean, what would that look like? And the idea is that they would look really different because those places are really far away from each other and there are no intervening native populations. So you would expect that it's as if I picked 10 people in this room and I sent you far away over to the divinity school and you stayed there for the next 500 years. Whatever genetic variants accumulated over at the divinity school would stay there and would be really different from whatever genetic variants accumulated in this room. So that was our null hypothesis, and fortunately there were some data for other Amanita species that are native to both places that showed this pattern. And that's not the pattern that we found at all. So here, yellow is representing California, blue is Europe, and gray is the East Coast. I just stuck in some of those specimens for fun. And every locus, every gene that we use to try to look at the genetic signature of the different populations shows that it's a muddle. And that's just another example of that. And I know the slide is really hard to read, so I'll tell you the story. The green here is a haplotype. The green here is a haplotype or a genetic type that's only found in southern Europe and not in northern Europe. This blue one is a genetic type that's only found in northern Europe and not southern Europe. But both of these unique types are found in California, strongly suggestive of multiple introductions from different parts of Europe to California. You can find, because these mushrooms, these fungi are diploid, you can find even in a single mushroom, so even in a single diploid individual, that part of its DNA comes from North Europe and part of its DNA comes from South Europe. So it really suggests a lot of mixing has happened. Okay, so with these data, we conclude that phalloides was introduced to California, and it's now the only known invasive mycorrhizal fungus really anywhere. And this is about the point when the NPR interview happened, and they came, and my daughter was 2 years old at the time. They came and interviewed her about whether or not she liked mushrooms. So on all things considered, if you listen to the story, you can hear my daughter saying, "I hate mushrooms." It's really funny. Okay, so Amanita phalloides becomes a unique case, but then we actually started digging in the literature to try to say that it seems...
Are there any genes in California that aren't in Europe?
No, none. That's a great question, which is another really strong piece of evidence. There was nothing unique to California that we found.
But there's stuff in Europe that's not in California?
Yes. Even though our sample size from California was lower as compared... our sample size from Europe was half our sample size in California. And even though, so our sample size was half in Europe, but there was much more genetic diversity in Europe than there was in California, which suggests also a bottleneck. Like not everything that's in Europe made it over to California. So this is just a slide to illustrate that since sort of we did the initial work on phalloides, we dug in the literature, and I mean we really dug in the literature. Elsa speaks Dutch and Italian and Spanish and French and some other languages. So we were literally reading like the bulletin of the Florentine Mycological Association for people who would say, gosh, we found this mushroom for the first time and we've never seen it in Florence before, to try to get a sense of how many introductions might have happened. And so we made this database. And the gray means we have no data. It doesn't mean that there aren't any introductions or any invasive species there. It just means that if I think mycology doesn't have enough people working on it in North America, boy, the situation in Africa is really amazing. There's hardly anyone working with fungi along here. And the green represents the number of introductions in any country. Again, this is just to show you that this has happened more than once. You can't really take quantitative data away from this because, for example, in New Zealand, they really care about what's been introduced to New Zealand. And so they keep careful track, and they know a lot about the fungi that have been introduced there. Okay, so how far has it spread and how quickly is it traveling? To do this work, I actually had to use herbarium specimens. I had to go to places where they keep dried plants and mushrooms. And I wanted to know in California, I got everything that was labeled as an Amanita phalloides because I wanted to know where this mushroom had first appeared and where it had gone. And so this just shows you that, at least initially, if you looked at those data, the early records were all from the south, and then later records that appeared more in the north. But you can't trust these for all the reasons that I've just said. And so what I had to do is I had to actually use my genetic tools to do what's called, annotate the specimens. I had to look at a specimen and literally write an annotation about what the specimen is as opposed to what someone else had said that it is. But I did this using DNA. And so these are all the species that for various reasons I wanted to compare phalloides to these because they're closely related. I wanted to compare phalloides to these because these are kinds of things that people want to eat. And sometimes when they pick up muscaria and they want to eat it because they want to get high, instead what they're eating is a phalloides. So they do take a trip, but it's not to the place where they thought they were going to take a trip. So this is the marker that I used to do this work. And this just shows you how this works. So this is like the actual DNA sequence, right? ATCG. And you can see, for example, that this is a phalloides that we have, and it has a G here. And that distinguishes it, for example, from this Ocreata, which is a closely related species, but doesn't have that G here. And this is just illustrated. We actually have a much longer stretch of DNA than I'm showing you here. And we've filled in a lot of the ends since then. Okay, so, and this also shows you that this locus, this gene, is invariant. There's no variability across the range of Amanita phalloides, but so these are specimens from all over the world, and there's no differences, but it's very different from what turns out to be the most closely related species to phalloides, which is Amanita ocreata. So it's a good marker. It doesn't vary within the species, but it's different between phalloides and all these other ones. All right, so then you can do something like you can go to this record collected in Santa Clara County in 1912, New York Botanical Garden, and actually it came up when it came up as not within our database at all. But fortunately, there are public databases of sequence data, and it turns out that this specimen is an Amanita pantherina. And the collector of this specimen did something sort of unusual. He deposited a photo of the mushroom. It's really rare. And actually, if you look at the description even, it doesn't sound like a phalloides. It has a cream-colored cap, which phalloides, I told you before, had a green cap. So in this case, I didn't need the DNA as much as I did for other specimens, for example, but I formally did the ancient DNA work for all of the herbarium specimens. So these are a lot of the specimens that I annotated using these ancient DNA techniques, the molecular marker. And what I want to show you is that all of these early records are not phalloides, even though they were labeled as phalloides. So this is just a different kind of evidence that phalloides wasn't around in the habitat before a certain time, but it also gives me a sort of a very poor fossil record about when phalloides first appeared in California. 1938 in Monterey and 1945 in Alameda. 1945 Alameda, this is the campus of the University of California at Berkeley, which prided itself on being a living forestry museum. So it brought in plants from all around the world to plant on the campus so that students of forestry could walk around and say, oh, that's a Canary Island pine. That's native to the Canary Islands, but here it is on the Berkeley campus and I can see what it looks like. Seemed like a good idea at the time. But inadvertently what they did was also introduce, I mean, when you put a plant in your garden and you take out that root ball, you have to think about all that stuff that you're putting in your ground in addition to the plant itself. Everything that's in that everything that's in that soil ecosystem, you're also putting into the ground. And that's exactly what happened here. The other record, this is the Naval Postgraduate School in Monterey, but it used to be a very fancy hotel called the Del Monte. And when it was a fancy hotel, 2 women mycologists visited it and they found Amanita phalloides in the shrubbery, that's what they say. So I got my French postdoc was not allowed to come with me on this trip, but I got special dispense because it's this Navy facility and it was just after, what did the French do? The French did something to make us angry, remember freedom fries and all that? So he wasn't allowed to come. But I was allowed to go. And this is a coast live oak, which is the main host for phalloides in California. And so it just is illustrative that here's the host that it jumped to, and here's where it was originally collected. So it wasn't, and these coast live oaks live for hundreds of years. So it's not hard to imagine it getting about that way. So here are the original data. After the molecular annotation, basically we've lost a lot because most of them weren't phalloides, but we have a current distribution. And so sort of combining all that information, you can see that it was originally found in these 2 counties in blue. By the 1970s, it had reached these counties in yellow, and it's currently in all the counties that are black. So that's certainly a pattern of spread. Yeah?
So when you were looking at the older issues or the ones that were collected earlier, and you were checking out the DNA to find out that they were different species, did you get a chance to see what they look like? Did they look morphologically like you anticipated they would, or? Yeah,
so that's a great question. So honestly, no. They didn't, but there's so much that you lose. Like when I find a phalloides, my main characteristics are color. I really want to see that sort of true phalloides color. And when you dry a mushroom, it just gets brown. And so you can't really look at that and say, oh, it's definitely, I mean, you can, the best that I can do is get it to genus in the sense that I'm really, really confident about getting a mushroom that I'm looking at to a genus, but getting it to a species is not trivial when you're looking at a dried specimen.
So does that mean that there could be phalloides that are in the collections that were misidentified originally also? So actually, if you would have been looking for one of the others, you might be finding? It's
definitely a possibility. And that, yes, it's absolutely a possibility. So, and what I think about that is that sort of in the sort of having to choose problems and how you choose problems. You couldn't chase after all of them. I also think that there's a bias towards labeling things as phalloides. So I think that's just sort of the human bias. People want to find phalloides and they label things that way. But that was also, your question is exactly why I couldn't just rely on the herbarium data to prove that it was an invasive species introduced from Europe. And the genetic data had to be the clincher on that. When I first started doing the work, I really thought, okay, I'm going to dig in the historical record and that's going to tell me what I need to know. And it turned out that it couldn't really exactly because of this question that you're asking. There's too much scope for human error in lots of different directions on that. Yeah. Okay, so so we've come along. I'm telling you a story that started in 2003, I think, is when I started this work. So the story has actually come a long way. And this is a more modern view of where we know phalloides is. And I'm using this just to show you the arrows are the historical introductions that I talked about. And this is a better picture of the current range of phalloides in California. This on the east coast those are just isolated arrows mark historical populations. And then the reason that they're just circles here is because it's not spreading on the east coast. So it's on the east coast, but it's not invading our local native forests in New England in the way that it's invading those beautiful coast live oak forests in California. And why is like the $1,000 million question. And we're looking into it, but I don't have an answer for you. But it's clearly doing something very different on the East Coast versus the West Coast. I mean, I could tell you all the interesting hypotheses. One of my favorite hypotheses is that the local diversity of Amanita species on the East Coast is really very much higher than the diversity of Amanita species on the West Coast. So along that line of thinking, you might think there's more room for it to invade in California because there just aren't as many conspecifics. There's not as much competition. The world Amanita expert, by the way, is a retired engineer from Bell Labs, and they bought him out. They offered him an early retirement package, and so they bought him out, and I think they even gave him a cash buyout. They must have, and he converted his garage into a herbarium, and he spent the last 40 years of his life just exclusively studying Amanita species. He lives in New Jersey, and when you go to his house, you literally go to his house, then you walk into his climate-controlled garage, where he has the world's largest, most valuable collection of Amanita species, and he's the recognized world expert. It's a fun story. His name is Rod Toulos, and he runs the Amanita. If you Google Amanita, his website will be one of the first ones that comes up. Okay, so this slide is basically what we did, so now we're sort of exploring many things. Okay, so we established phalloides as an introduction. Now we want to know why is it invasive in some places and not in others, and where's it going, and who is it associating with, and many different kinds of things like that. So here's a map of North America, obviously. What we've done here is we've taken what we know about this fungus in Europe. So we have lots of places where we know it grows in Europe, and from those European locations, we can understand something about temperature, where it grows, something about rainfall, where it grows in Europe. And then we can say, understanding its habitat in Europe, we can project onto a map of North America where we think suitable habitat is in North America. And that's what you're looking at here. And the more white it is, the higher the probability of finding phalloides there. And red marks known populations of phalloides. So you can see, for example, that Mexico lights up like a Christmas tree. So we don't think phalloides is there yet. There aren't any documented collections there. But based on this, we can give this kind of science some predictive power. We can say that if phalloides is introduced to Mexico, chances that it will establish there are high. And you can see, for example, that actually in general, the West Coast is a more suitable place than the East Coast. Also the Ohio River Valley. So now we have a huge network of amateurs that work with us to try to describe and document this mushroom, and we definitely have people who are on the lookout in this area also. We thought we had a collection from Ohio last year, but we didn't. So people think that a rough mean for plants to get around is about 2 kilometers a year, and for phalloides, based on, in particular, the herbarium data, where it was and where we know it is now, we calculate that it's spreading about 10 kilometers a year. These data are pretty rough, but it gives you an idea. Okay, so mechanisms of a range expansion. That's a story about funding in science, because when I tried to get funding, one of the things that people kept wanting me to tell them is how to get rid of phalloides from a habitat. And I wasn't, and so someone suggested to me that I should do an experiment where I have one plot where I burn it and another plot where I don't burn it, and sort of monitor that over time and see whether burning makes phalloides go away. It's a ridiculous suggestion, but along those lines. But in the course of answering a totally different question, I did discover that actually phalloides gets around exclusively using its spores. In other words, every fungus has the capacity for asexual propagation. So you can tear apart any fungus and it'll regrow from both halves. So there was a question about whether it was getting around with spores or whether it was getting around just using chunks of itself underground. So through the course of this work, I found that spores is the answer. And because I know that, I can tell people who want to get rid of phalloides on their landscape that, and I also know from this work that it's very short-lived. An individual doesn't live for more than about a couple years, 2 years maybe. That if you wanted to rid your yard of phalloides, which is a perfectly legitimate concern if you have children or dogs, then all you need to do is patrol and pick every mushroom before the gills open. And if you do that for several years, you actually will rid your habitat of phalloides. So I did discover a way to get rid of it. I think it shows you the value of basic, and I'm probably preaching to the choir here, but there is a genuine value to basic research as opposed to directed applied research, which is also can be incredibly valuable, but anyway, funding basic research is a public good. Yeah?
So what are you supposed to do with them once you rip them up?
Just throw them in the trash.
And that's enough?
Yeah. Yeah, that should be fine. Yeah. You don't need to do nothing, yeah, so it's nothing more complicated than that. Okay, so who's phalloides associating with? And this is the last sort of research bit. So in California, we find phalloides in these really beautiful undisturbed forests. In New Jersey, we find it in these plantations. So these are actually plantations that were planted by the Civilian Conservation Corps in the 1930s. It is a native host, it's eastern white pine, but for some reason in the middle of this beautiful New Jersey pine barrens habitat, a chunk of it was ripped out and they planted this eastern white pine. And you always find phalloides within the plantation, and you never find phalloides in the native vegetation, and we have been looking. It doesn't grow there. So it's a question of what, of proving what hosts it's associating with in California versus here, and figuring out how that plays out in terms of the current distribution of phalloides. So how we do that is we actually build a library of DNA sequences from above ground tissues. So pine DNA looks really different from oak DNA. And then we go to the root tip. Remember, the root tip is the site of the physical site of the symbiosis between the fungus and the plant. And we get the fungal DNA out to confirm that we're collecting phalloides, so we've got the right target species. And then we get the root DNA out because, and then we match it. So in this case, you can see it lights up the same color as the pine. So we would say, from having isolated this root tip from the soil, that in this case we have a phalloides associating with a pine and not an oak. And what we see when we do that is on the East Coast, remember I told you it's not invasive, it's not spreading around, it's really associating with pines. Whereas in Europe, it's really associating with things, with oak things, which is what I've been telling you from the start, but this is a really nice set of data. This is a really nice set of data to show you that it's oak. And then we also just did some comparative work to look at its host associations in Europe. So the associations on the West Coast are mirroring its associations in Europe, its native habitat, whereas on the East Coast it's not quite the same thing. All right, and we know that this oak that we're looking at, you're looking here at a map of Point Reyes National Seashore. So San Francisco is here, and Berkeley, California is right over here. And if you look there at particular sites that are field sites, you can see that Quercus agrifolia, the coast live oak, which I've shown you is the dominant host, or is the dominant host in these mixed stands. So a part of the point, so I showed you a map where we had projected European abiotic variables like temperature and rainfall onto North America to see what suitable habitat is. So there are certainly abiotic controls on where phalloides is going and where it's spreading, but then these data also make the point that because it's a symbiotic fungus, it's not just temperature and precipitation that are going to shape its range. It's also what compatible hosts are available in the local habitat that will shape its range. So here you're looking at gray are just areas where there's so this is land, this is ocean, the same outline you saw before. Red are where we know phalloides grows, and you can see that the red more or less matches the green, which is Quercus agrifolia, which is the dominant host. So all of this, all right, so I started off talking about what we don't know about biodiversity and that there's a great deal of work left to be done when we're describing fungal species. I illustrated that there is such a thing as an invasive fungus that's non-pathogenic. We know things like chestnut blight, right, and Dutch elm disease because we don't have chestnuts or elms anymore by and large. But a lot of fungi are not pathogenic, and so there's a whole world of biodiversity that isn't out there killing our plants, and that's the world where I live in. So there's a lot of it. There's a lot of it in our local habitat, and there's a lot that we don't know, but we know that things like invasions are happening, so there's a sense maybe that we might be losing things before we have names for them. And that's also the world where I live, and I think about that a lot. What kind of biodiversity loss are we experiencing that we don't know about? So I just want to end with one more story, just a story to tell that, to say that same thing again. So Amanita inopinata is an Amanita species, a mushroom, that was first described by a guy named Reed. He discovered it in the UK, and he recorded it as an introduction to the UK because he'd never seen it before. Inopinata means the unexpected one in Latin. So that's the unexpected Amanita. He was surprised to see it. So a little bit later on, this Field Mycology is a journal that a lot of amateurs and professionals participate in, but it's basically just like a fun, fun mycology journal. And they have fungal portraits every time they publish. And so Amanita inopinata was given by Geoffrey Kibbe this fungal portrait. And so he said, here it is, it's growing in the UK. And this is a really distinct, it's not exactly a pretty mushroom, but it's a very distinctive mushroom. You would know it if you saw it. And he said, it's growing in the UK, and it was introduced to the UK from New Zealand, is what he said. Well, in the very next issue of Field Mycology, a different Geoffrey Ridley, said, hey, he said, as to where it comes from, I do not consider it native to New Zealand. Yes, it grows in New Zealand, but it's not native there, and speculate that it comes from Europe. Okay, well, you can speculate away. Meanwhile, the unexpected one is traveling in Europe. It's invading. And so Kees Boas writes this wonderful article, also in Field Mycology, where he discovers that Amanita phalloides has jumped across the North Sea and now is on continental Europe as well as the UK. And he says, yes, I said Amanita inopinata, but that species is known only from New Zealand and southeastern England. Are the spores amyloid? That's a character that you use to discuss it. So then he decides that the spores are amyloid, so it's really an Amanita inopinata. My heart really made a little jump, and I immediately recognized it as Amanita inopinata, not least because of the color photographs published this year in Field Mycology. This was really an exciting discovery. I nearly kissed the finder, but instead we danced a bit around the table. I live in a good world, mycologists. So here we have a situation where we have the unexpected one, where the people in Europe are saying it's introduced to us, and the people in New Zealand are saying it's introduced to us, but nobody knows really where it's introduced from. And I put a big question mark in Africa because there's so much we don't know about what grows there. Although I'm sticking to my theme of we don't know our local biodiversity particularly well either. So I titled the talk, Last Chance to Know, because I do think that it is our last chance to know something about the world as it existed before we entered the Anthropocene. So I don't know how many people have heard that word before, but I really like it. The Anthropocene suggests an era where humans are the dominant force. And if someone comes at you and tells you that they're an ecologist and they work at some field station and they're studying something that has nothing to do with humans, I'm here to tell you it's a lie. I don't think that any aspect of our natural world has escaped human influence. And so I think if we care, and I don't know if we care, I don't think it's a question for scientists. I think all evidence points to we don't care. I don't see many of us changing our habits, despite what we all know. I suspect that this is a particularly educated audience, and because I'm in New England, I suspect that many people have a particular point of view on conservation biology, also because people are biologists. But I would also suspect that in this room, there are very few of us who have decided to take mass transit instead of driving to work every day. We make active choices in our lives. from what I can tell, those active choices do not point to saving biodiversity. I don't see that we're going to stop that loss. So I think we're at an edge, and I think that every day when I study what I study, that we're at the end of being able to know something about the world as it was. And then we move forward, and my daughters will move forward in a changed world. If you haven't read this book, that's where my title comes from. It's by this guy, Douglas Adams, who wrote The Hitchhiker's Guide to the Galaxy. You used to be able to use that as a reference for your students. You probably can't anymore. And he went around the world and he looked at species that are on the brink of extinction, and he recorded what he saw about them. So that's where the title comes from.
Yeah, I guess I'm wondering, invasive species, usually we consider them invasive because they're harming something that was there before. Do you have any evidence of what may have been the local fungi and how they're impacted?
No, it's because we have no baseline data. So when you don't have baseline data, something shifts in and it's around and it's dominant, but you have no data to say what was there before versus what's there now. So molecular biology has really revolutionized our study of fungi. The PCR machine has made it possible for us to see things in a way that we never saw them before, which is why mycology is such a vibrant field right now. But nonetheless, phalloides is there. It's invasive. There's no really good controlled experiment I can do to go to a place where it is and say, and I can't match it with a place that doesn't have phalloides and say, this is what it looked like before. But I want to talk also about the definition of invasive species. So my definition I understand perfectly well why you say that. Clinton himself actually published a bulletin about invasive species where he invoked this idea of harm as part of the definition of an invasive species. I use a more neutral, I use the word more neutrally. So an invasive species to me is one that had a range, and then humans picked it up and moved it to a new range, and it's spreading, and then I leave harm out of it. But you could probably get 20 people in a room who do invasion biology and ask them, and you would come up with, it's very frustrating, 20 different definitions of what an invasive species is.
Do you have a word then that means that they're causing harm?
No, I don't. And I think that's just something that you might, I think the word invasive, the word invasive comes from Charles Elton, who coined the word after World War II. And if you read his original book that really established invasion biology, it's filled with the language of battles and warfare. And we have not escaped that language. Scientists as humans haven't escaped that language. So I guess usually what I do is leave it alone. I did, I think, at some point in one of my writings define how I use the word invasive, but then other than that, I've just let it go. Yeah. Yeah.
The Quercus agrifolia in California, is it very similar to the oaks in Europe?
It's a great question. So actually, yes, but there's also a species in New England or in Northeast North America that's also very closely related to the European species. So it's not that the California oak is more closely related to the European species Quercus robra than the New England one is. That's not it. There's something else going on. It's not a phylogenetic constraint. Yeah. But it's definitely something we thought about. Yeah.
Is this coast live oak, is that invasive?
No, it's native to California. It's usually they're very old forest stands. They're, no. It's an endemic to California. And it's not planted, really. Sometimes in people's gardens, they like to plant it. Yeah. Yeah.
So it's probably because you didn't get a chance to talk about the distribution and where it was going, but you inferred that they were spread by sexual spores, and yet we're transplanting or we're moving them. So what is it that we're actually moving?
The mycelium in soil. So when we pick up a seedling, a plant seedling, and it's in a pot, what you're actually moving in that soil is a living fungal body. That's called the mycelium. And then you put it into the ground and it grows, and it probably rapidly forms a mushroom and then spreads spores.
So then it's not that they can't spread or increase the distribution of where they're at through asexual means, because
Correct.
They must be able to do that.
They can.
It's just that it's significantly slower, and maybe that's not how they would.
Yeah, and I think it needs a human in order for them to be able to do that. It needs a human to pick up the soil and the body, the mycelium of the fungus, and move it to a different place in order for that to work. Naturally on their own, unless a human does that, naturally on their own, they would get around with spores. And most spores fall very close to the mushroom. Most spores just go right by the mushroom. Yeah. Yeah, there's one way in the back.
I know you have these maps of abiotic factors as far as temperature and precipitation, but
there's lots of maps in this country, one of the few things I seem to find maps on is soil texture and soil science. Is there some sort of overlapping with that?
Yeah, it's a great question. I left it out because, I don't know, basically to make the story just a little less complicated. But that was also a part of what we looked at in that it's called a species distribution model. So we also looked at soil, features of the soil. So it does play a role. Yeah.
I had two kind of side comments. One was I had a student ask me about fairy rings.
Oh yeah.
And I had no idea. I was like, something about mycelium growing that way would create them in a circle. I was wondering if you have an answer on that.
Sure.
And my other question was about what actually causes the death cap to kill you. It said something about amino acids on there.
Oh, yeah. Okay, so to answer your first question, when a fungus grows, you can imagine it, there's a spore, and it creates a mycelium, right? And then I'm just going to start to draw it as a ring. As time goes by, it gets bigger and bigger, right? It grows in a sort of, in all directions, in a circular front. Does that make sense? And then it's only the leading edge that makes mushrooms. So in year 1, you would find mushrooms here in a circle. And then in year 2, you would find mushrooms here in a circle. So actually, you can infer the age of a fungal individual by how big the fairy ring is if you know something about its growth rate. Yeah. Now, this is not a symbiotic fungus. This is a decomposer fungus. And we didn't talk about it at all today, but obviously another reason that fungi are huge in the environment is because they control flows of carbon. So terrestrial soils are a huge sink of carbon, and it's really the decomposer fungi that are the biomass and doing the work of breaking apart plants and that kind of stuff. And fairy rings are characteristic of that. Some mycorrhizal species do form them, but yeah. And then your second question. Okay, so there are 6 toxins in Amanita phalloides. There are the amanitins and the phallotoxins. And it's the amanitins that actually kill people. So what happens is that you eat a mushroom and you vomit and you have diarrhea about maybe, 4 to 6 hours after you eat the mushroom. Then you feel better, and you often feel better for a period of about 10 hours or so. Then you go right back into vomiting and diarrhea, and it's usually at that point that the patient presents at a hospital, which is a little bit problematic because sometimes they don't mention that they've eaten a wild mushroom because it seems like so much time has elapsed. It's a pretty violent, horrible death. So what happens is that the toxins are absorbed in your blood, and it's the liver that filters them out. And in particular, I think it's the alpha amanitin that stops your RNA polymerase II from working. And so that shuts down the function of your liver, and that's why the older remedy for Amanita phalloides poisonings is a liver transplant. And then there's lots of complications associated with liver transplants. As probably a lot of you know, you have to be on immunosuppressive therapies your entire life after you've had a transplant. But this new therapy seems to work really well, actually. They put this new therapy that's derived from a milk thistle plant, a European milk thistle plant. They put you on palliative care. So in other words, they pump you full of liquids, and they take care of you very well and give you this milk thistle at the same time, and it seems to negate the need for a liver transplant. All right, thanks very much.
Thank you.